Learning goals. Welcome to this learning session on mitosis, stages and characteristics of cell division. In this course, we shall address Introduction to Mitosis and Cell Basics. Mitosis stages overview and interphase. Prophase, condensation, metaphase, alignment. Anaphase, separation. Telophase, reformation. Cytokinesis and mitosis significance.
Introduction to cell division. Cell division is a fundamental biological process by which a parent cell divides into two or more daughter cells. It is essential for growth, repair, and reproduction in all living organisms. There are two primary types of cell division, mitosis and meiosis. Mitosis is the focus of this course, primarily responsible for the proliferation of somatic body cells. Meiosis, on the other hand, is a specialized form of cell division that produces gametes, or sex cells, and will be covered in a separate lesson. Defining mitosis and body cells. Mitosis is a type of cell division that occurs in somatic, or body cells. These are any cells in an organism that do not produce gametes, such as skin cells, muscle cells, and blood cells. This process is crucial for the growth and development of multicellular organisms. The primary purpose of mitosis is to produce new cells for growth, replace old or damaged cells, and facilitate asexual reproduction in some organisms. For instance, your body replaces millions of skin cells every day through mitosis. Parent and daughter cells. During mitosis, the original cell that undergoes division is referred to as the parent cell. This parent cell meticulously duplicates its genetic material before dividing. The two new cells formed as a result of this division are called daughter cells. A defining characteristic of mitosis is that the daughter cells are genetically identical to the parent cell. This means they contain the exact same number and type of chromosomes. This genetic fidelity is vital for maintaining consistent cellular function and tissue integrity throughout an organism's life. The diploid state in mitosis. A crucial aspect of mitosis is that if the parent cell is diploid, the daughter cells will also be diploid. Diploid 2N means that a cell contains two complete sets of chromosomes, one set inherited from each parent. For humans, this means 46 chromosomes arranged in 23 pairs. This maintenance of the diploid number ensures that all somatic cells in an organism have the full complement of genetic information. For example, a skin cell will have the same number of chromosomes as a liver cell, both being diploid and genetically identical to the original zygote from which the organism developed. Mitosis is a continuous process, but for ease of study, scientists divide it into several distinct stages. These stages ensure the orderly distribution of genetic material to the daughter cells. A helpful mnemonic to remember the order of these stages is IMAT. IMAT stands for interphase, prophase, metaphase, anaphase, and telophase. While interphase is technically a preparatory phase before mitosis begins, it is crucial for understanding the entire cell cycle. Cytokinesis, the final division of the cytoplasm, often overlaps with telophase. Interphase, cell preparation. Interphase is the longest phase of the cell cycle, during which the cell grows, carries out normal metabolic functions, and prepares for division. It is divided into three subphases: G1, growth; S, DNA synthesis; and G2, further growth and preparation for mitosis. Crucially, during the S phase, the cell's DNA is replicated, meaning each chromosome is duplicated to form two identical sister chromatids. These sister chromatids remain joined at a constricted region called the centromere. At the end of interphase, the nuclear membrane is still intact, but chromosomes begin to condense and become visible under a microscope. Prophase, chromosome condensation. Prophase marks the beginning of active mitosis. During this stage, the replicated chromosomes, each consisting of two sister chromatids, condense and become much shorter and thicker, making them visible as distinct structures under a light microscope. This condensation is crucial for preventing tangling during chromosome segregation. Simultaneously, the nuclear envelope begins to break down into small vesicles, and the nucleolus disappears. The mitotic spindle, composed of microtubules, starts to form between the centrosomes, which move to opposite poles of the cell. This spindle will be responsible for separating the chromosomes. Metaphase, chromosome alignment. Metaphase is characterized by the alignment of all the condensed chromosomes along the metaphase plate, also known as the equatorial plate, which is an imaginary plane equidistant from the two spindle poles. This precise alignment is critical for ensuring that each daughter cell receives an identical set of chromosomes. During this stage, each chromosome's centromere is attached to spindle microtubules from opposite poles. The tension created by these opposing forces ensures that the chromosomes are perfectly positioned before separation. This stage is often the easiest to visualize under a microscope due to the distinct alignment. Anaphase, sister chromatid separation. Anaphase is a rapid and dramatic stage where the sister chromatids suddenly separate from each other. The centromeres divide, and each chromatid, now considered an individual chromosome, begins to move towards opposite poles of the cell. This separation is driven by the shortening of the spindle microtubules. As the chromosomes are pulled apart, the cell temporarily elongates, contributing to the separation of the genetic material. By the end of anaphase, an identical set of chromosomes has arrived at each pole, ensuring that the future daughter cells will be genetically identical. Telophase, nuclear reformation. Telophase is essentially the reverse of prophase. Once the chromosomes have reached their respective poles, they begin to decondense, returning to their more diffuse chromatin state. A new nuclear envelope forms around each set of chromosomes, creating two distinct nuclei within the single parent cell. The mitotic spindle fibers disappear, and the nucleoli reappear within the newly formed nuclei. At this point, the cell contains two complete, identical sets of chromosomes, each enclosed within its own nucleus, preparing for the final step of cell division. Cytokinesis, final cell division. Cytokinesis is the final stage of cell division, where the cytoplasm of the parent cell divides to form two separate daughter cells. This process typically begins during late anaphase or telophase and concludes shortly after telophase. In animal cells, cytokinesis involves the formation of a cleavage furrow, an indentation in the cell surface that deepens until the cell is pinched in two. This furrow is formed by a contractile ring of actin and myosin filaments. In plant cells, a cell plate forms in the middle of the cell, growing outwards until it fuses with the existing cell wall, creating two new cells. Significance of mitosis. Mitosis is a fundamental process with profound biological significance. It is the basis for growth in multicellular organisms, allowing a single-celled zygote to develop into a complex organism with trillions of cells. Without mitosis, organisms would not be able to increase in size or develop specialized tissues. Furthermore, mitosis is essential for tissue repair and regeneration. When tissues are damaged, such as from a cut or burn, mitosis produces new cells to replace the lost ones, ensuring the integrity and function of organs. It also plays a role in asexual reproduction, where a single parent organism produces genetically identical offspring, like in many plants and single-celled organisms.
Summary. Well done. You have completed the course, Mitosis, Stages and Characteristics of Cell Division. You should now be familiar with Introduction to Mitosis and Cell Basics, Mitosis Stages, Overview and Interphase, Prophase, Condensation, Metaphase, Alignment, anaphase, separation, telophase, reformation, cytokinesis, and mitosis significance. Should you need to revisit any of the topics in this course, please use the menu at the bottom of the screen.